We're going to finish up photosynthesis in this lecture. So we've been talking about the photosystems, and there's actually two different photosystems. There's photosystem 2, which is used first, and then there's photosystem 1, which is used second. So they're actually named in the order that they were discovered, so that gets a little confusing. The photosystem 2 has two special chlorophyll A molecules in their reaction center, and they're called P680 after the wavelength of light that they absorb the best. So they absorb the 680 nanometer wavelength of light best, which is in the red range. And then photosystem 1, the reaction center there, the chlorophyll A molecules are called P700 because they absorb the 700 nanometer wavelength of light the best. And between photosystem 2 and photosystem 1, there's an electron transport chain. So this would be important to remember. Photosystem 2 is going to produce ATP, and photosystem 1 is what produces the NADPH and the light-dependent reactions. And we're just going to look at the non-cyclic electron flow, and you don't have to worry about cyclic electron flow at all. So in non-cyclic electron flow, we're just saying that electrons are just going to flow one way through the photosystem, so they're not going to circle back at all. So if we start with our light energy hitting our photosystem 2, gets absorbed by the chlorophyll, and they're going to pass around that light energy until it gets to the reaction center complex on photosystem 2. That'll boost the electrons up in the P680 special chlorophyll A molecules, and then those electrons are going to go down that electron transport chain. And then those electrons will end up at the reaction center complex of photosystem 1. And along the way, as the electrons travel down the electron transport chain, they're going to release energy, and that energy is going to be used to make ATP. So you can see that photosystem 2 is going to be used to make ATP in the light-dependent reactions. So the electrons, when they get to P700, they're low energy now, so we need to boost them up with more energy. So that's going to come from light energy. And the light energy gets passed around until it gets to the reaction center complex, boosts those electrons up, goes down a second electron transport chain, and then those electrons are going to be put onto our electron carrier, and that's where the NADPH is made. So you can see that photosystem 1 makes NADPH. Okay, and then we need to answer this really important question here. So I would star this in your notes. So from where are the electrons that are lost from P680 and P700 replaced? So it's just saying these electrons that we boosted up in photosystem 2 and in photosystem 1, where do we replace them from? And so if we look at our picture, you can see that we're going to get electrons from water. So we're going to split the water apart, take the electrons off water, and then those will get boosted up by the light energy, go down the electron transport chain, be boosted up by more light energy, and then finally ended up on the NADPH. And then in the process of breaking apart the water, we make the oxygen as our waste product. So to answer our question, from where are these electrons replaced? They're replaced from water. So that would be good to know. Okay, and then this is all explained here, but there's a picture that has everything with the light-dependent reactions. So we've got, this is just showing you a eukaryotic microorganism. We've got chloroplasts, and this would be one of the thylakoids, little green discs here. And here are the photosystems, photosystem 2, photosystem 1. And you can see the light energy hitting photosystem 2, boosting electrons up, going down the first electron transport chain, and then those electrons being boosted up again, and then ending up on NADPH. And then remember that those electrons are replaced from breaking apart water, and we make oxygen as a waste product. And as those electrons go down that first electron transport chain, the energy that's released from those electrons is going to be used to pump hydrogen ions. And those hydrogen ions are going to create a concentration gradient. So we end up with lots of hydrogen ions inside this thylakoid. And notice that we have some ATP synthase. Remember that the mitochondria have those as well. And so we end up creating, with this hydrogen ion concentration gradient, a proton motive force. Because remember, hydrogen ions and protons are the same thing. And the hydrogen ions or protons are going to move down through the ATP synthase. That'll cause it to spin. And that'll provide the energy to shove the phosphate group onto ADP to make ATP. So that's where the ATP comes from. And so we've made both the NADPH and ATP that are made by the light-dependent reactions, and those will be used by the Calvin cycle. Okay, and then speaking of the Calvin cycle, that's our second step of photosynthesis. So this is where we're going to fix carbon dioxide. We're going to make it part of an organic molecule, and we're going to use that to make glucose. And I would know where this occurs in both eukaryotes and prokaryotes. So in eukaryotic microorganisms, Calvin cycle is going to occur in the stroma of the chloroplast. And in prokaryotes, because they don't have chloroplasts, it'll just occur in their cytoplasm. Okay, so this is showing you a chloroplast that would be in a eukaryotic microorganism like algae. And you can see that the Calvin cycle would be out here in the stroma. So kind of this gel-like material that's outside the thylakoids. Okay, so important things to know about the Calvin cycle. We're going to be fixing carbon dioxide, and that just means that you're going to make it part of an organic molecule. And the enzyme that is able to fix carbon dioxide is rubisco. So we don't have this enzyme, so we cannot fix carbon dioxide. So we have to rely on photosynthetic organisms to do this for us, and this is where we get all of our carbon from. And you can also see that the Calvin cycle requires energy, ATP, and also some electrons from our electron carrier NADPH. So in all of this, ATP and NADPH is all coming from the light-dependent reactions. Okay, and this last part is just kind of a summary. So to answer this first question, what does the cell do with the ATP produced from breaking down food? So when food is broken down, all of those chemical bonds that are broken, they release energy, and that energy will be temporarily stored as ATP. Well, that ATP will then be used by the cell to do work later. So whether it's going to be chemical work, transport work, or maybe mechanical work. Okay, and then our next question, which electron carrier is used by catabolic reactions and is used to make ATP? So this would be NADH. Those electrons carried by NADH will go to the electron transport chain, get dumped off there, and that'll be used to make ATP. Okay, and then we'll look at this next thing in the middle here, how we can pull off things, intermediates, to use to make other things in anabolic reactions. So we'll look at that in just a minute. Okay, but our next thing, which electron carrier is used for anabolic reactions? Okay, so remember this is NADPH. So these electrons, instead of being taken to electron transport chain, are going to be used in anabolic reaction. Okay, so I would definitely remember what NADH is used for and NADPH. And then lastly, what types of organic molecules does the cell need to make? So all four of them. So carbohydrates, lipids, proteins, and nucleic acids. And all this picture is just showing you is that from aerobic respiration pathways, you can see that the different intermediates can be pulled off and used to make different things that the body can use. So you don't need to memorize any of this, but you can just look at it. Okay, and then there's a lot of different chemical cycles that your book talks about, but I just wanted to just talk about one of them, just the nitrogen cycle. So let's think about which organic compounds contain nitrogen. So hopefully you remembered that we talked about how proteins and nucleic acids both contain nitrogen. Because remember, proteins contain amino acids which have nitrogen in them, and then nucleic acids, the nucleotides, have a nitrogenous base in them. So the nitrogen cycle depends on bacteria to happen. So it's going to start with nitrogen fixation, which is where we've got some different types of bacteria. We talked about the rhizobium bacteria, which live in the root nodules of the legume plants. And also cyanobacteria can also do nitrogen fixation as well. But what they're able to do is they will take nitrogen gas that's in the air and they will convert it into ammonia. So NH3. And then that can be used to make organic molecules. Oops, ran out of room. Okay, and then because it's a cycle, we need to then eventually return our nitrogen back to nitrogen gas. So from nitrogen fixation, we then go to nitrification, which that's defined as we convert the ammonia to nitrites or nitrates. And I'm not going to ask you these chemical formulas. So okay, and then denitrification. D is talking about removing. So we're going to be removing the nitrogen, which means we're going to be putting it back into the air. So we're just going to convert the nitrates to nitrogen gas. And now we're back to where we started again. We're back to the nitrogen gas. Just a nice picture, just showing you the nitrogen gas in the air, which we can't do anything with. And then nitrogen fixation, here we've got a legume plant, maybe we've got some rhizobium bacteria, and they're able to convert the nitrogen gas into the ammonia, which then can be used to make some organic molecules like proteins, nucleic acids. And then nitrification is where the ammonia gets converted into the nitrates and nitrites. And then denitrification is where the nitrates get converted into nitrogen gas, and then we're back where we started again. So just knowing that we've got this nitrogen cycle and just you've got these 3 steps there.